Hey all, so I apologize in advance. I think this one might be a long one. We're going to be looking at pages 413 to 427, and we're looking at C2.1, which is chemical signaling for HL students. So we're trying to think about how do cells distinguish between the chemical signals they receive and what interactions happen inside animal cells in response to chemical signals. And we're thinking about interaction and interdependence here. So there are receptors on cells, and signals can be sent via chemicals, which would be made in one cell and then bind selectively to a receptor site on another cell. And that binding molecule is going to be called a ligand. So it's similar to an enzyme substrate specificity. Enzymes and receptors do not permanently change shape with the binding, and it's going to be specific. Receptors receive the signals, they don't change the chemical structure of the ligand, whereas enzymes do change the chemical structure of their substrate. But a ligand can be bound for a long time. And what's happening is that the sensor is going to send a signal to the cell while that ligand is bound. And what we can have is when we have a sufficient population of bacteria, they can do something called quorum sensing, which is where they're all signaling similarly, and that's going to generate a chemical reaction. They send a chemical signal and it's going to generate a reaction from all, and that's going to change their gene expression. So bioluminescence in certain organisms can be caused by bacteria. So for example, in squids like this picture here, it's bacteria in their light organ, and it's going to affect the camouflage of the squid. And so what's happening there is there's a high enough population of bacteria in that light organ. They send autoinducer signals, and that's going to trigger the production of luciferase, and that's going to create a glow. In the case of bioluminescence that we see in the ocean, that's being caused by algae. So that's working through a different process. So with hormones, we have hormones and neurotransmitters. Hormones are chemical messages that are secreted by the glands, and they travel in the bloodstream through the whole body to the target cells, and they will up or down regulate the action of the target cells, so make it happen more or make it happen less. And they can last in the body for many hours and act on many cells. So examples of hormones might be thyroxine, testosterone, or insulin. Insulin we've already learned about. And then neurotransmitters are chemicals that are released via the presynaptic neuron, going to the postsynaptic neuron or to an effector. And they're going to travel across that synaptic gap to the postsynaptic neuron, either exciting it or inhibiting it. So there's a very short distance traveled here. Their binding opens channels and ions move in by facilitated diffusion, and that's going to change the membrane potentials. Neurotransmitters are broken down or reuptaken quickly, such as dopamine, which is uptaken, and acetylcholine, which is broken down. So you can see hormones and neurotransmitters, their action pathway is quite different. Then we also have some other signaling cytokines. These are produced by many different types of cells, but their effect is short range. They're interacting with nearby cells or their own cells, and these are related to allergic reactions. So you can see this would be a cytokine signal. These signals are binding to receptors on cell surfaces, leading to changes in gene expression, and that's going to be related to inflammation and the immune system. And so examples of cytokines are interferon, interleukin, and erythropoietin. And then calcium ions are signals in neurons and muscles, and they're happening with presynaptic neurons. We've already talked about this, where calcium channels open to trigger the release of neurotransmitters from vesicles into the synaptic gap, and they are then pumped back out of the gap into the presynaptic neuron. In muscles, calcium is stored in the sarcoplasmic reticulum. And when muscles receive a signal from a motor neuron, the calcium is released from the sarcoplasmic reticulum, and that's going to bind onto a protein called troponin, which reveals tropomyosin, and that's going to allow muscle contraction to happen. And then calcium is pumped back into the sarcoplasmic reticulum. So troponin, if it's not bound up with calcium, prevents the contraction of muscles. And that's what this picture is here. So that's the sarcoplasmic reticulum, and calcium here would be binding onto troponin. So we have endocrine glands. We've already talked about exocrine glands. Those are more related to our digestive system. We saw how our pancreas is both an endocrine gland and an exocrine gland because it's related to juices for digestion, but then also it's an endocrine gland because it's releasing insulin and glucagon. And so endocrine glands secrete hormones directly into the bloodstream. So an example of this would be thyroxine. There are three types of hormones. There's steroid hormones, protein hormones, and amine hormones. And they're going to bind to receptors to trigger a sequence of reactions called a signal transduction pathway. But we're going to look at specifically steroid and protein hormones and how those interact differently in our cells. So in steroid hormones, because they are a type of lipid, they can pass through the cell membrane and join with receptors inside the cell. And the receptor hormone complex is going to promote or inhibit directly transcription of a gene. So you can see it's linked here. This is the hormone receptor complex, and it is going to produce messenger RNA. And that is going to mean that that protein is produced. So examples of steroid hormones would be estrogen, cortisol, calciferol, progesterone, or testosterone. And then in the steroid hormone interaction, we have intracellular receptors. And so those receptors in here, so you can see the receptor protein right there, have hydrophilic amino acids, and so they're going to remain dissolved in the cytoplasm of that cell. So for example, testosterone increases the FAD-S1 gene, which then increases the amount of fat for prostate cells. So that would be the action of testosterone as an endocrine hormone. And then progesterone and estrogen, also very common steroid hormones. And so before and during ovulation, where a woman releases an egg from her ovaries, estradiol from the ovaries signals to the receptors in the brain, in the hypothalamus, to release GnRH, which stands for gonadotropin releasing hormone. And gonadotropin releasing hormone is going to signal the release of luteinizing hormone and follicle stimulating hormone from the anterior pituitary gland. And then those hormones are going to act on the ovaries to release progesterone. And that's going to maintain the uterine lining. So it's going to do so by increasing, this FSH and LH are going to trigger ovulation. And then progesterone separately as a hormone, steroid hormone from the ovary, is going to maintain the uterine lining. And it does so by increasing insulin-like growth factor receptors in the uterus, which is going to promote cell proliferation. So more and more cells grow inside the uterus. So then protein hormones are working a little bit differently. Peptide hormones attach to receptors on the cell membrane, triggering secondary messengers in the cell. And so one very common one is CAMP. So ATP is modified into the secondary messenger called CAMP. And these receptor proteins are called transmembrane receptors that have a band of hydrophobic amino acids that are attracted to the nonpolar tails of the phospholipid membrane. And then there are hydrophilic amino acids that are in contact with the aqueous solution, both inside and outside the cell. And so that receptor binds onto it, and it's going to trigger this secondary messenger process, which we'll look at it in a bit more detail in the next slide. Amine hormones action is not always the same, and it sometimes acts like peptide hormones, and they sometimes act as steroid hormones. That's the most that you need to know about that. So transmembrane receptors, the G protein complex is an example of this. So G protein coupled receptors are a large and diverse group of transmembrane receptors, and they send signals via a secondary protein called the G protein. So it has 3 subunits, alpha, beta, gamma, on the receptor. And I think this image is probably a little, maybe it's this one here. Let's look at this one here. So we have our G, alpha, beta, gamma, and initially G, alpha, beta, gamma, GDP is attached here to the G alpha. And so that's GDP is guanosine diphosphate. It's bound to that alpha subunit. When a ligand binds, then that GDP is going to detach and it's going to allow GTP to bind instead. And so once the GTP binds, it's going to detach the subunits from the receptor. So the alpha subunit stays there, but beta and gamma go off and do their own thing. And it allows them to facilitate the secondary messenger production. And so we have GTP here has interacted, and then we have ATP moving to make CAMP that is going to allow, this is what's happening when epinephrine is binding onto a transmembrane receptor. So we have CAMP happening, and that's going to activate some different components that will then break down glycogen into glucose in the liver. Additionally, and so we actually saw this here. This is looking at different kinds of kinases. So tyrosine kinase activity, kinase is an enzyme that adds a phosphate from ATP to a molecule. And when insulin receptors are activated by insulin, the 2 tails of the receptor, which are the tyrosine kinase, they're going to attach to each other. And in that attachment process, they're going to add ATP to each other. And this is going to trigger vesicles containing glucose transporters to come to the membrane and insert transporters into the membrane so that, and this is all triggered by insulin, that's that little orange blob up there, so that glucose can now enter into the cell. And so all of these can have hormone signaling, can be affecting positive and or negative feedback loops. So we've already talked about positive feedback loops, and that's the end product of a pathway amplifies the starting point. So more product is made or a new equilibrium is achieved. And so in the case of childbirth, a drop in progesterone is going to activate oxytocin production in women. And then oxytocin is going to cause uterine contractions. And then the more contractions that the person has, the more release of oxytocin. And when I say women, I should say people with uteruses. And then a negative feedback loop is going to shut off the pathway. And so, for example, GnRH from the hypothalamus, remember that stands for gonadotropin releasing hormone from the hypothalamus, triggers the production of LH. And LH is actually important not only for ovulation, but for triggering the Leydig cells in the testes to produce testosterone. And so as the amount of testosterone signal increases, it's going to stop LH and then also stop GnRH from interacting with the Leydig cells. So to summarize, chemical signaling can occur through a variety of pathways. It could happen with ligands, neurotransmitters, hormones, and cytokines. And endocrine glands release, and additionally ions here, like calcium ions. Endocrine glands release hormones into the blood, and 2 main types of hormones are peptide and steroid, which have different action pathways, ligand binding versus to transmembrane versus intracellular receptors. And these hormone interactions can act in positive and negative feedback loops. So neurotransmitters are released from neurons and have shorter action time and smaller action area than hormones do. All right, that's it for this video. Thank you for watching.